Hey everyone, let's jump right into this explainer and explore the fascinating world of high-fidelity DNA replication. Today, I'm putting on my molecular biologist hat to guide you through a step-by-step masterclass on how our cells copy their genetic material. Now, instead of just looking at a bunch of abstract chemical reactions, we're going to treat the replisome, which is the massive protein complex responsible for all this, like a highly coordinated, dynamic factory floor. It's an absolute marvel of precision engineering, and I can't wait to show you exactly how it works. Here's our roadmap for today's masterclass. We'll start with Section 1, the factory floor blueprint. Move to Section 2, unzipping the DNA helix. Then, Section 3, the molecular assembly line, followed by Section 4, quality control and proofreading, and we'll wrap up with Section 5, prokaryotes versus eukaryotes. All right, Section 1, the factory floor blueprint. Let's take a look at the foundational design of DNA replication. To really understand our molecular factory, we first need to look at its foundational blueprint. Back in 1953, Watson and Crick proposed the double helix structure, but it was the famous Mendelssohn-Stahl experiment in 1958 that proved exactly how it copies itself. They confirmed what we call the semi-conservative mechanism. This simply means that when DNA replicates, the original double helix splits, and each half serves as a master template. So every new daughter cell inherits a DNA molecule composed of one original parent strand and one freshly synthesized strand. It's basically the ultimate biological backup system. Now, because DNA is a massive molecule, the factory doesn't just start at one end and slowly chug along to the other. That would take way too long. Instead, replication starts at specific sequences called origins of replication. The DNA opens up to form a replication bubble, and expanding outward in both directions, you get these Y-shaped regions called replication forks. This right here is the active site. It's the very edge of our factory floor where our industrial machinery actually operates to duplicate the code. Moving on to Section 2, unzipping the DNA helix. Let's see how the factory prepares the template strands for the assembly line. Before we can build, we have to deconstruct. Right at the replication fork, we have our industrial unzipper, an enzyme called helicase. It acts as a motor protein that aggressively breaks the hydrogen bonds holding the two complementary DNA strands together. But there's a catch. Those single strands naturally want to snap right back together. So to stop this, the factory deploys single-strand binding proteins, or SSBs for short. They coat the exposed DNA, acting as scaffolds to prevent the strands from prematurely reannealing while the rest of the machinery gets to work. Now, as helicase aggressively unzips the DNA, it creates an intense mechanical problem. Imagine pulling apart two heavily braided ropes. The tension builds up further down the line, right? Well, in DNA, this causes severe supercoiling ahead of the replication fork. If left unchecked, the whole factory would just grind to a halt. Enter topoisomerase, an absolutely essential tension-relieving enzyme. It literally cuts the phosphate backbone of the DNA, lets it untwist to release the stress, and then seals it back together. This ensures the helicase can keep unzipping without tying the DNA into impossible knots. Let's do a quick checkpoint to test your understanding of this first mechanical step before we bring in the master builders. Think back a couple of steps. What specialized proteins coat and stabilize the exposed DNA once helicase breaks it apart? If you said single-strand binding proteins, or SSBs, you are absolutely right. Great job! Section 3, the molecular assembly line. Time to start building the new DNA strands. With our DNA successfully unzipped and stabilized, it's time to fire up the precision manufacturing process. Our master builder here is an enzyme called DNA polymerase. But there's a major biological quirk. DNA polymerase can only add new nucleotides in one specific direction, from the 5' end to the 3' end. And because our two original DNA strands are anti-parallel, meaning they run in opposite directions, the factory has to elegantly solve this by building the two new strands differently. The leading strand is built continuously, just following the helicase right into the fork. But the opposite side, the lagging strand, has to be built backward, away from the fork, in these short, discontinuous bursts we call Okazaki fragments. Let's zoom in on how those lagging strand Okazaki fragments are actually manufactured. See, DNA polymerase can't just start building out of thin air. It needs a starting block. So step 1, an enzyme called RNA primase lays down a short RNA primer. Step 2, DNA polymerase uses that primer to synthesize a short stretch of DNA. That's your Okazaki fragment. Finally, after specific DNA repair factors swoop in to remove those temporary RNA primers, step 3 happens. An enzyme called DNA ligase comes in acting like molecular glue, sealing the gaps between the fragments with really strong phosphodiester bonds. This beautifully organized procedure ensures that even the discontinuous lagging strand is stitched together perfectly into a solid, intact DNA molecule. Our replosome is a truly dynamic, bustling factory floor. It's definitely not just polymerase working all alone. To keep the polymerase from falling off the DNA track, a concept we refer to as processivity, specialized clamp-loading factors attach ring-shaped sliding clamp proteins right around the DNA. These clamps act like safety tethers, anchoring the high-fidelity polymerases firmly to the template so they can seamlessly perform their rapid synthesis without constantly falling off and having to reattach. Let's pause for another quick checkpoint to verify your grasp of this asymmetrical elongation machinery. Do you remember what we call those short pieces of DNA built backward on the lagging side? They are called Okazaki fragments, named after the incredible pioneering scientists who first discovered them. Section 4, Quality Control and Proofreading. Let's look at how the factory prevents catastrophic manufacturing errors. We all know even the best factories make mistakes, which brings us to a topic I am deeply passionate about: the strict quality control mechanisms that are absolutely essential for our genomic stability. A staggering fact is that our DNA polymerases are incredibly precise. Through their innate ability to select the correct building blocks, combined with their active proofreading, they typically only leave behind one wrong nucleotide for every 100 million to 10 billion base pairs copied. Given that our genome is billions of letters long, this incredible fidelity is literally what keeps us alive and healthy. But how exactly does it fix an error when one inevitably slips through? Well, the polymerase essentially has a built-in proofreader. Step 1. If it inserts the wrong base, it immediately senses a physical distortion in the geometry of the DNA double helix. Step 2. The enzyme actually hits the brakes and physically moves the DNA strand about 30 to 40 angstroms away from the polymerase active site and into a distinct exonuclease active site. Step 3. This 3' to 5' exonuclease acts as an editor, chopping out the incorrect nucleotide. The strand is then safely shuttled back to the main site to resume proper building. So, why does this matter so much? Well, when this microscopic proofreading machinery fails, the consequences are severe. If errors bypass that 3' to 5' exonuclease domain, the resulting faulty DNA causes rapid genomic instability. In the clinical world, we see that mutations crippling the specific exonuclease editor in our cellular polymerases are directly linked to a massive increase in mutation rates. This mutator phenotype is a root cause of severe human diseases, and it's strongly associated with various sporadic and hereditary cancers. Take a moment to recall the exact name of this vital proofreading function before we look at the broader evolutionary picture. What is the editor called? It's the 3' to 5' exonuclease activity. Just remember that directionality. It's moving backward to fix the mistake. Finally, Section 5, Prokaryotes versus Eukaryotes. Let's see how evolutionary adaptations shaped the replication factory across different domains of life. Prokaryotic and eukaryotic factories have vastly different kinetics and logistics. As you can see here, prokaryotes are built for blistering speed out in the cytoplasm, adding a whopping 2,000 nucleotides per second. Eukaryotes, on the other hand, are way slower and more methodical. Operating safely inside the nucleus, our polymerases only add about 100 nucleotides per second. Why the big difference? Because eukaryotic replication forks must navigate a dense, complex obstacle course of chromatin structure and nucleosomes that bacteria simply don't have to deal with. There's also a major architectural difference. Prokaryotes have a small circular genome. They really only need a single origin of replication, just one replication bubble that goes all the way around the circle. Eukaryotes, however, have massive linear chromosomes. Because our polymerases move so much slower through that dense chromatin, if we only had one starting point, it would take weeks to replicate our DNA. Instead, eukaryotic replication features hundreds to thousands of simultaneous origin sites, all firing at the exact same time to get the job done efficiently. Now, that linear eukaryotic structure creates a very unique dilemma, known as the end replication problem. When the replication fork reaches the very end of our linear chromosomes, there's just no room left to place a final RNA primer on the lagging strand. Conventional polymerases simply cannot finish the very tips. If left alone, our chromosomes would shrink with every single cell division, eventually leading to cell death. Thankfully, an incredible specialized enzyme called telomerase swoops in to save the day. It acts as an extension machine, using its own built-in RNA template to extend these ends, which we call telomeres, protecting our genome from that progressive shortening. Ultimately, the structural conservation of the replisome proves that nature found a brilliant mechanical solution to replication incredibly early on. The fundamental tools, like helicases, single-strand binding proteins, and polymerases, are remarkably similar across the board. They address universal replication challenges, adapting perfectly whether it's for blistering speed in a simple bacterial cytoplasm or operating under the highly evolved regulatory checkpoints required within the incredibly complex environment of a human cell nucleus. I want to leave you with this final challenge to ponder. If we understand exactly how this DNA factory works, from topoisomerase relieving tension all the way to the delicate balance of the sliding clamps, how might we use that knowledge clinically? Could we design targeted drugs to selectively jam the gears of the replication line in rapidly dividing cancer cells without harming our healthy factories? It's a massive question driving the very forefront of molecular biology today. Thank you so much for joining me on this masterclass. Keep questioning, keep learning, and I'll see you in the next explainer.